Shri Gurubhyo Namaha children. I welcome you all to our online teaching academy Ekalaiva. Today in this video we are going to see how to separate our DNA fragments which has been made by restriction enzymes. Okay. In our last video we learnt about the restriction enzyme action. Okay. We saw what are the restriction enzymes and how they are acting on our DNA to make them into a fragments. Okay. Now you have your DNA fragments in your hand and we are going to see how to separate those DNA fragments. Okay. And for separating these DNA fragments, we are using a technique called gel electrophoresis. Okay. Gel electrophoresis. Okay. In this, on this gel, okay, we can separate our DNA fragments according to their size. Okay. We will see that in detail. Okay. So, to make this the gel electrophoretic unit, okay, you need to prepare an agarose gel. Okay. So, this restriction enzymes, okay, you have seen that in your last video, the restriction enzymes prepared our DNA fragments because they have their own restriction sites in our DNA. So, when you use that restriction enzyme, it will go and cut in that restriction sites. So, we will be getting the fragments of our DNA. So, we have our DNA and you all know that our DNA is having the charge, okay, negative charge. So, using this charge of DNA, we are going to separate our DNA fragments, okay, under the electric field. Okay, so that process only we will call it as electrophoresis, and this electrophoresis we are going to do it using a gel called agarose gel. Okay, and this technique we are calling it as a gel electrophoresis. Okay, so now the medium what we are going to use here is agarose gel, as I told you, and this agarose gel, it is a natural polymer which has been extracted from seaweeds. Okay. So, using that agarose gel, we are going to make our electrophoretic unit. Okay. So, this agarose gel, what is the use? Why we are using this agarose gel? When you see the sieving effect of agarose gel, okay, when this agarose gel gets solidified, okay, it will be having some pores inside it. Okay. So, this sieving effect, it will help our DNA fragments to move according to their size. Because of that only, in gel electrophoresis, we are using this agarose gel. Okay. So, we will see how to prepare this unit. Actually, this is our electrophoretic unit. Okay. The apparatus what we are using for this electrophoresis, and this is our gel caster, okay, where we are going to pour our agarose gel, and this is called comb. Okay. So, this is gel caster where you are going to pour your agarose gel, and this is comb. Okay. So, after inserting this comb, actually you will prepare agarose gel and it is heated to dissolve that agarose completely in that buffer. Okay. Actually agarose is dissolved in the buffer and it is heated for the complete dissolution. So, once it is dissolved, you have to pour it in this gel caster. Okay. So, you have to pour it in the gel caster. So, set up the gel caster with a well comb. I showed you how the comb is inserted. Okay. So, make sure that the ends of caster trays, okay, all the four sides should be sealed properly, okay, or else agarose when you are pouring it will come out. Okay. So, to avoid that, all the four sides has to be sealed properly. Okay, then you have to insert the comb, then the prepared agarose, okay, without the formation of air bubbles, okay, without the formation of air bubbles, it has to be poured. Okay. So, this warm agarose, it is poured slowly to prevent bubble formation. If bubble formation is there, it will be affecting the sieving property of this agarose. Okay. Then after pouring it, allow it to solidify for 20 to 30 minutes without disturbing it. Okay. So, after that time when you see, okay, this agarose gel will undergo solidification. So, you can see two pictures here. Okay, this one at once after you are pouring that agarose gel in the gel caster. So, that is before solidification. Once it is solidified, the gel becomes translucent like this. Okay. So, by seeing the translucent gel, you can know that it is solidified. So, you can remove that comb. Okay. The plastic comb what you have inserted before pouring the agarose gel can be removed. Okay, so when you remove, you can see, okay, the wells are made here for loading our DNA sample. Okay, for loading our DNA sample. So that is the purpose why you are inserting your comb in this gel electrophoresis. Okay, so now our gel is ready now for electrophoresis. So what you have to do now we are having our DNA sample and our electrophoretic unit is ready. So now the sample is added into the wells using this micropipette. Okay, using this micropipette, actually it is added into this gel. Okay, gel wells. So once you have added, okay, it is connected with our cathode and anode, and okay, you switch on this electrophoretic unit. Okay, once it is switched on, the current flowing from cathode to anode. So you all know that, okay, you should be very clear about this concept. Being DNA is a negatively charged substance, you know that. So being negatively charged, the sample well has to be made near this cathode. Okay, so you know that, okay, opposite charges repel. So being a negatively charged particle, the DNA will start moving towards this anode. So for making that movement only, okay, you are making your sample well near the cathode end. So now the electrophoresis started. Now, okay, this DNA fragment started moving on this gel. Okay, very clearly you can see from the well. See how this blue color things you are seeing, no? That is actually showing the path how our DNA sample started moving through this agarose gel. I told you already, agarose gel is having the sieving effect is there. So due to that, according to their size, the DNA fragments get separated. Okay, so this is how it started. Okay, so you have to wait, and once this DNA sample reach that anode end, you have to switch off that electrophoretic unit, or else our DNA fragments will fall into the buffer what we are using. So once it is reaching this end of anode end, okay, you have to switch off the electrophoretic unit, then you can collect our gel. Okay, this is the gel where the DNA fragments has been separated. Only the stains are visible here. Okay, you can see the sample, actually along with the sample we have added a blue color stain. Okay, that stain color alone you can see here. Okay, you don't know how many DNA fragments are there, how many bands of DNA formed. So how to visualize the DNA? Okay, it is very easy. How you are visualizing your DNA when you see? By using a special stain. Okay, so that stain is called ethidium bromide. Okay, you're going to stain the gel, okay, which has been subjected to electrophoresis. It is stained with a special type of stain called ethidium bromide. Okay, you have to be very careful when you are handling this ethidium bromide stain, because it is carcinogenic in nature. Okay, so this gel, okay, it will be taken in a gel box and it is added with this ethidium bromide stain. Okay, it will stay. Okay, you can stain it for few minutes. Okay, after that you have to take the gel, and this apparatus is called UV illuminator. UV illuminator. Okay, UV light source will be there. Okay, so when you keep your gel in this UV illuminator, you can see orange color bands. See, you can see bands. Yes, here some bands are there, and here some bands are there. Okay, so according to their size, okay, to see precisely according to their molecular weight, the DNA fragments started moving towards anode. The heavier fragments will stand very near to the well. Okay, the smaller one will move away from the well because the size is small, so that they can move for a long distance. Okay, so this is how our DNA fragments can be separated using this agarose gel. Now you want that particular DNA fragment from the gel. How you can separate the DNA, particular fragment of DNA from that gel when you see? It is by using the process called elution. Okay, how to prepare, how to separate when you see that particular part of gel. Okay, here you are having a various bands are there. Now we are going to separate this particular DNA fragments. So what you have to do now? Okay, you have to cut this particular piece of that gel, agarose gel. Okay, the particular piece of agarose gel, it has to be cut from that whole electrophoresis gel, and it is taken in the buffer like this. It is dissolved in the buffer, then it is subjected to centrifugation. Okay, it is subjected to centrifugation. So when you centrifuge, okay, you can get your DNA in the form of precipitate. Okay, and this DNA, it is eluted with a buffer called tris EDTA buffer. Now, okay, the particular fragment of DNA is isolated, okay, it is separated successfully using this gel electrophoresis method. So this is how you can separate, you can make the DNA fragments using your restriction enzyme, and you can separate that particular fragment using your gel electrophoresis. To receive our online lessons, please press the subscribe button and you will receive the latest updates. Thank you.